This is a production of Cornell University.
Okay, thank you, Kevin. I'm going to continue our discussion of Cucurbita pepo and talk about the work that I've been doing looking at resistance to the disease Phytophthora crown and root rot. So the causal agent of this disease is Phytophthora capsaceae, which infects a wide variety of plants, including many economically important vegetable crops, such as most solanaceous species, all cucurbits, and some legumes as well. Phytophthora capsaceae can infect all plant organs and causes different symptoms, the most important of which I'm showing on this slide. We see fruit rot on the left. The white growth that you see is the pathogen sporulating. And I'm focusing on crown and root rot, symptoms of which we see on the right. Crown and root rot causes dampening off symptoms in young seedlings, but also affects more mature plants, causing lesions on the roots and stems, which lead to wilting and plant death. So in pepper, there are sources of very high disease resistance that are used in breeding, and there's been a lot of work looking at the genetics of disease resistance, but much less is known in Cucurbitaceae. Although many accessions of many cucurbit species have been screened for either fruit rot resistance or root and crown rot resistance, and there's a wide amount of variation in terms of degree of susceptibility, although there are very few cases of clear complete resistance to the pathogen. So my work is focused on Cucurbita pepo, which, as we learned from Warren, includes zucchini and pumpkin, among other market classes of squash. And in Cucurbita pepo, there are no sources of complete resistance to this disease that have been identified. And most cultivars are highly susceptible to Phytophthora root and crown rot. So here at Cornell in the Mazurek lab, in recent years, there's been work to develop pumpkin and zucchini lines with an increased amount of resistance to Phytophthora crown and root rot. And the most promising lines, which I'm working with for my research, were derived from a cross between zucchini cultivar Romulus and a gray zucchini landrace from the National Plant Germplasm System. In the picture, we can see on the left one of the advanced lines compared to susceptible zucchini variety Dunja. And these advanced lines are partially resistant in that the disease progresses more slowly and symptoms are reduced, but we still observe plant mortality on many plants of these genotypes. However, they perform better than the top accessions from a previous study that evaluated several hundred Cucurbita pepo germplasm accessions. I've also challenged these breeding lines with 7 diverse pathogen isolates, and the response to disease is consistent, indicating the absence of any isolate-specific interactions. So one of the goals of my research is to identify loci that control resistance to Phytophthora crown and root rot, and to do that, I've developed an F2 mapping population. We can see the parents and the F1 on the left. The susceptible parent is zucchini variety Dunja. In the middle, we have the breeding line that I previously identified as being most resistant. And on the right is the F1. The figure on the right shows area under the disease progress curve on the parents in the F1. Area under the disease progress curve is a measurement of disease progression over time. In this case, using measurements of disease incidence every other day for 2 to 3 weeks following inoculation. We can see that the F1 is not intermediate between the parents, but is closer in phenotype to the susceptible parent. And we can also see that the differences between the parents are robust to differences in inoculum concentration, where even inoculating the partially resistant parent with a suspension of 100,000 zoes per mil, we observe less disease than on the susceptible parent inoculated with a suspension of 1,000 zoes per mil. So we expect the genetic architecture of this trait to be complex. And with that in mind, we are employing bulk-segregant analysis with whole genome resequencing of the bulks to map QTL. The idea behind this is straightforward. It consists of evaluating a large number of plants identifying the phenotypic extremes, in this case, the most susceptible and the most resistant, sequencing those DNA pools from those bulks, estimating allele frequencies genome-wide, and identifying regions that differ in allele frequency between the bulks. So there can be several advantages in some cases over traditional QTL mapping where all individuals are phenotyped and genotyped separately. That is because you're genotyping fewer individuals, genotyping costs are lesser. Phenotyping is also simpler in the sense that instead of assigning a precise phenotypic score to all individuals, it just requires the researcher to identify the phenotypic extremes. There can be a statistical challenge in separating signal in allele frequency differences between bulks from sampling noise from sequencing, but several methods have been developed that increase the accuracy of allele frequency estimates at SNPs by borrowing information from neighboring markers. So this slide shows how I implemented this approach in the F2 population I mentioned earlier. I evaluated over 13,000 F2 seedlings by spraying them with a suspension of Phytophthora zoospores, and identified bulks, not just a susceptible and resistant bulk, but also a random bulk to estimate baseline allele frequencies in the population. And I attempted to have the bulks constitute 15% of the population. Selection was applied in each 72 cell tray that the plants were sowed in, so that corresponds to 11 plants in each tray being selected for the bulks. And then tissue from those 11 plants was bulked, DNA was extracted, and I'm currently quantifying it and standardizing across 96 trays to end up with 6 DNA pools, the 3 bulks from both stages of phenotyping that will shortly be submitted for sequencing. I'm also interested in the mechanisms that allow plants of these partially resistant genotypes to have a higher probability of survival compared to other genotypes. And previous work has shown that structural and physical features in roots and stems are involved in resistance to Phytophthora crown and root rot. And a previous experiment using peppers showed that Phytophthora zoospores were able to attach to and germinate equally on roots of both susceptible and resistant peppers. But on susceptible peppers, the pathogen was able to penetrate into the interior of roots, whereas on resistant genotypes, growth was limited to the outer cell layers of the cortex. So this summer, with the help of an undergraduate intern who worked on this project, we used a green fluorescent protein tagged isolate and confocal laser scanning microscopy to look at the infection process in both one of our partially resistant breeding lines, 17G009, and the susceptible zucchini variety Dunja. We looked at 96 taproot cross sections and gave them a binary score of hyphae present or absent in the root interiors and in the vascular tissue. And both 3 days post-inoculation and 6 days post-inoculation, we observed a higher probability of observing hyphae in either the root interior or the vascular tissue in our susceptible variety Dunja. But we still observed hyphae in a substantial portion of the partially resistant breeding lines. And I think this is interesting because whereas in the greenhouse, we observed almost all of the Dunja plants to be dead by 7 days post-inoculation, by a couple of weeks post-inoculation, we only observed 10 to 20% of the partially resistant breeding line to be dead, although we observed many more of the plants to have the pathogen present in the interior of the root. So it seems that both penetration of the pathogen is limited in the partially resistant genotype, and that the plant's able to continue to grow and survive despite infection for a period of time compared to the susceptible variety. So my next steps are, I'm currently developing additional mapping populations that I will use to validate any QTL that I'm able to detect in the Bulk-segregant experiment and to estimate their effects. And I also hope to continue to do some additional microscopy experiments and differential gene expression analyses to further understand what makes a resistant plant more resistant. So with that, I'd like to thank the SMART and Core Labs, the other members of my committee, Michael Mazurk and Philip Messer, Kyle LaPlante in the Mazurk Lab, who I collaborate with on these projects, technician in the SMART Lab, Holly Lang, Ella Reeves was the undergraduate who worked on the microscopy experiments, and funding from the New York State Department of Agriculture and Markets and the USDA National Institute of Food and Agriculture. Thank you.
Questions for Greg?
Yeah.
So since PCAP doesn't have an airborne form, it seems like you would have more diversification, or like the isolates in Illinois would just be completely different from those in New Jersey versus something where the spores can spread 1,000 miles. I mean, are there lots of issues of basically race-specific resistance like being an issue to PCAP?
There's evidence in pepper for race-specific resistance. There's not much evidence in cucurbits. I looked at the response of these partially resistant lines, mostly to isolates from New York, but also a couple of isolates from the West Coast, and the response was the same in all cases. Yeah, Mark.
You said that some of the resistant plants died and some didn't. Do you have any idea what the difference is in those plants? Was there more hyphae, or could you tell just what the cause was?
I don't know. In part, I think there's kind of maybe a threshold in terms of infection, and there's variation in terms of how many plants of the same genotype reach that threshold and end up dying, and we see that in pepper as well. But I'm interested in maybe using quantitative PCR to try to estimate pathogen abundance and associate that with survival.
If there's no more questions, let's thank Greg and all the other speakers again.
This has been a production of Cornell University, on the web at cornell.edu.